If you had followed from the start the signal transduction pathways, we had discussed that signal transduction has a specific ways of giving the information to the cells. So basically, signal transduction mechanisms are the means of interacting with the cells with the surroundings. So chemical messengers here are nothing but like hormones, and they are going to bind to the particular receptors if they are proteinaceous, polar. If they are non-polar, steroidal means they can directly cross the cell membrane. So this way, these chemical messengers, depending on their nature of the um, character, they can either pass the signals from the cell membrane through the receptors, or they can directly cross if they are lipid-based molecules. Then there are different stages. So this is the reception, then execution, then effect. So different stages mainly of any signal transduction pathways. So the final effector molecules, whatever may be the signal transduction pathway as we discussed, those are going to be transcription factors, and these transcription factors will translocate into the nucleus and responsible for the expression of proteins that have got role in that particular cell physiology. So second messengers now, these are the small molecular substances that are mediating the upstream receptors after their activation and downstream effector molecules. So there are different type of second messengers. So CAMP we discussed that itself is a second messenger, then calcium is another second messenger in the cell. Likewise, even lipid-based phosphoinositol derivatives, those are also acting as a second messengers. These are the small molecular substances that mediate the upstream signaling event and downstream effector molecules. They are in between, the mediators. Okay. So in that way, partly we had discussed, but in this topic in detail, we are going to discuss about the second messengers. So today's class mainly about the general properties of these second messengers, then functions and formation of intracellular messengers, and then with the examples, CAMP and other examples, we are going to continue with the discussion. So the first topic is about the general properties of the second messengers. So signal transmission from the receptor to the interior of the cell takes place in two ways. If you see the different signal transduction pathways. One is protein-protein interactions. So different proteins of the signal transduction pathway interact with each other. That may be interaction of the receptor. If it's a proteinaceous hormone like insulin, it will interact with the receptor. Okay, so at the level of the receptors, these protein-protein interactions can be seen. Then inside the cell, the relay of the signal information is through protein kinases. Protein kinases again, they are nothing but the proteins. So one of the upstream protein kinase, when it gets activated, last chapter we discussed that serine threonine kinases, they are responsible for phosphorylation at the catalytic site where serine, threonine, tyrosine are there. You also know different classifications of these kinases, right? So these are the protein kinases. So once the upstream protein kinase got activated after its phosphorylation, it is responsible for activation of downstream protein kinase by phosphorylating. Then another downstream protein kinase operates. So likewise, a cascade will be formed, cascade of the pathway. And these are mainly because of protein kinase interactions, nothing but protein-protein interactions. Then other way of signal transmission is through the low molecular weight messengers. So through protein-protein interactions, and then the low molecular weight messengers, that is nothing but second messengers. So these are also like mediators that transmit or relay the signals from the receptor to the transcription factors. So the intracellular messengers are formed or released. They are either formed or released by specific enzyme reaction during the process of signal transduction, and they serve as an effectors by which the activity of proteins further is regulated. So basically, in that way, it is medial. They are getting activated after its release or formation as a byproduct of enzymatic reactions, and then effectors, the downstream effectors, will be activated by this second messenger. So this is, for example, the cell with different second messengers. Like here, DAG is another second messenger which is present in the membrane. You will have either in membrane or as a free form in the cytosol, second messengers. Calcium here, for example, is free form in the cell and DAG in the membrane. So these molecules, and here it is inositol triphosphate. So these are second messenger, and there are different messengers, second messengers, which are formed or released, will then activate the downstream effector molecules. Understood? Now, second messengers are the group of small molecules that mediate the relay of signaling events from the upstream receptors to the downstream effector molecules. Understood? So that's the way to explain second messengers. So the second messengers are diffusible signal molecules that can reach the target by diffusion. So there are different types, and if it is cytosolic form, they actually diffuse. By diffusion, they reach the target sites. So two types of these second messengers based on their nature, either hydrophobic or hydrophilic. So second messengers can be divided into two broad groups: hydrophilic and hydrophobic second messengers. Hydrophobic means non-polar, like hydrocarbons. Okay, hydrocarbon chain, fatty acids, they are non-polar, hydrophobic, lipid molecules. So these are like second messengers. Diacylglycerol, which is associated with the membrane. So membrane associated second messenger molecules are hydrophobic second messenger, because in the membrane is hydrophobic in nature. It has got lipid layer. So these type of second messengers that are associated with membranes, like diacylglycerol, phosphatidyl inositol derivatives. Okay, and these are localized in the membrane, and they are the hydrophobic second messengers. The main function of these types of messengers is the promotion of membrane localization of the target proteins. In the cytosol, if there are any proteins, they will be localized to membrane through the second messengers. That is done by this hydrophobic second messengers. Understood? Next is hydrophilic type. Hydrophilic means water philicity. So water itself is a polar molecule. So this will be a polar. So hydrophilic second messengers are polar charged molecules. And in the cytosol, where the water is the maximum that you have, it will localize through the process of diffusion. Simple diffusion is a process or means by which this hydrophilic second messengers in the cytosol can relocate to the site of the effector protein. So in many cases, these target protein means the one which is acted upon by the second messenger. Understood? So the target proteins are activated by an allosteric mechanism upon binding of the second messenger. Allosteric mechanisms you know very well. One, when it binds, it facilitates with an ease more velocity of other molecules to bind. Okay, that is the allosteric regulation. So this is the same way even second messenger's binding is taking place. That is hydrophilic second messenger. So the major effectors of second messengers. So second messenger activation will affect different protein kinases and also GF proteins, that is gene nucleotide exchange factors along with ion channels. So these are all the downstream molecules of the hydrophilic second messengers. And hydrophobic second messengers affects the target molecules which have to locate to the membrane. Understood all of you? This way, second messengers are mainly divided into hydrophobic and hydrophilic types of second messengers. So the second messengers can be formed and degraded again in specific enzyme reactions. The small secondary messengers are formed and degraded. This is one of the problems. Formation and degradation is continuous in the cell by a particular enzymatic reactions. So through enzyme-mediated pathways, the large amounts of messenger substances can be created and inactivated. So enzyme-dependent mechanisms are responsible for second messengers to form and to degrade in the cell. So like calcium, the second messengers can be stored in some specialized storage organelles. Calcium, for instance, in the cell is stored in the cell organelle called reticulum. Endoplasmic reticulum is the part where calcium storage is taking place. So messenger substances are produced in a location specific manner, and they may also be removed or inactivated according to their location. It is therefore possible for the cell to create signals that are spatially and temporally linked. So second messengers are under the spatio-temporal regulation. Now you know in signal transduction already we discussed spatio-temporal regulation means spatio that is with particular locality, temporal that with respect to particular condition, the time of the cell. Understood? So that way even second messengers are under the control. So these are the different properties of the second messengers. Any questions anybody who wanted to ask? Is anybody having any questions, clarifications, doubts?
No,
Okay. So if there are no questions, then next topic will move on with functions and formation of these intracellular messengers. So this is now here you see intracellular messengers. They are in the middle, mediator role in the signal transduction pathways. So here you can see this is serpentine receptors, your G protein coupled receptors, and this is receptor tyrosine kinases. When they get activated, different effector proteins can be mediating their effects of receptor activation, and this through effectors, the small group of molecules will be activated. And we discussed in the earlier slide that these second messengers which mediate the signaling pathway from the effectors to downstream effectors, that is of two types, either diffusible, so diffusible are polar molecules, and another one is membrane associated. So these are hydrophobic, that's why it can anchor with the membrane. Okay, so that's why these are hydrophobic lipid, lipophilic. Hydrophobic are lipophilic. Hydrophilic are lipophobic. So they are water soluble. Water is a polar molecule. So these are basically polar. So that's why these should be in the cytosol and easily it can diffuse. So diffusible messengers and membrane associated messengers. Hydrophilic and hydrophobic type of second messengers. So hydrophilic example, CAMP, CGMP, inositol triphosphate, then calcium. All these are in the cytosol and they reach the target site by diffusion. Whereas in the membrane, which are hydrophobic, these are DAG, diacylglycerol, phosphodiol 3-phosphate. This is inositol triphosphate. This is phosphatidol inositol triphosphate. There is a difference. Okay, this you need to appreciate. So I made this yellow border, and you should appreciate that this yellow border second messengers are like mediators in between the upstream receptor activated effectors and downstream protein kinases. Okay, and other molecules, downstream effectors. Understood? So now here membrane associated hydrophobic second messengers are responsible for activation of protein kinases, and these are also membrane bounds. So molecules which are associated within the membrane are activated mostly by these hydrophobic membrane associated messengers, whereas hydrophilic diffusible second messengers like CAMP, CGMP, calcium inositol triphosphate, they are responsible for activation of protein kinases, then release of the calcium, then ion channels that are getting opened. So these are all the different ways how the second messengers can mediate their effects. Understood all of you? So second messenger is basically two types, hydrophilic and hydrophobic. Hydrophobic are membrane associated and will be anchored to the membrane, whereas hydrophilic, they reach the target site through the simple process of the diffusion. Understood? So intracellular messengers in signaling pathway have critical roles as mediating upstream to downstream effectors. So they are like mediators, or I said broker. Brokers will be, very well. Brokers will mediate if you have any work in the government office. If you go through mediators, your work will be done faster, not at trauma ecology. Okay. So like that, second messengers are like the brokers in the cell. They mediate the effects of upstream signaling pathway to the downstream effector of the pathway. So hydrophilic messengers can diffuse in the cytosol as we discussed and activate the effector molecules, whereas hydrophobic messengers, in contrast, they remains in the cell membrane. Hydrophobic, lipophilic, so it has to be with membrane, and they can diffuse within the cell membrane. Within the cell membrane, they can diffuse so that it reaches the target protein. Understood? So now examples here, as already in the depiction mentioned, cytosolic second messengers like CAMP, CGMP, inositol, phosphate, and calcium, whereas membrane-associated second messengers, which are hydrophobic or lipophilic, like lipid derived molecules. Lipid-based signalings are through membrane-associated second messengers like diacylglycerol, phosphatidyl, inositol triphosphate. So that is the formation and functions of the signaling molecules. Then we are going to start with the examples of the second messengers, one after the other in your chapter. So first one is, you know very well, we had discussed the role of cyclic AMP. Earlier in the class, where did we discuss about the cyclic AMP? I think we had written even in the exam also. Somebody should tell. Where we discuss CAMP? Cyclic AMP, where did we discuss? Sanjana?
Sir, in protein kinase and adenylyl cyclase also.
Right, very good. Adenylyl cyclase is the enzyme which is responsible for the formation of CAMP. And another enzyme here you can note down. See here, phosphodiesterase. So CAMP is degraded after its function by phosphodiesterases. So these two enzymes you need to memorize. Adenylyl cyclase is the enzyme which is responsible for formation of 3' 5' cyclic AMP from the ATP substrate. Whereas after its function, CAMP undergoes degradation process by the action of phosphodiesterase. So in the cell, the maintenance of CAMP levels is very important because they are important second messengers, and they are responsible for triggering the downstream effector molecules of the pathway. So CAMP levels in that way very critical when it is required. Spatio-temporal regulation. The second messengers are under spatio-temporal regulation. Means depending on the condition or time, at what condition of the cell cycle or whatever it may be. Differentiation, cell cycle. These are the different time points of the cell. Okay. Now, spatio, that is location. Which location? So these two aspects are importantly regulating the second messengers. So CAMP is also in that way it is under the control. And the levels of CAMP is very important because whether CAMP mediated signaling is on or off depends on the concentration of CAMP in the cell. And that is maintained, the CAMP levels are maintained in the cell by the regulation of two important enzymes, that is the synthesizing enzyme CAMP synthesizing enzyme, that is adenylyl cyclase, and CAMP degradation enzyme, which is nothing but phosphodiesterase. So these two enzymes in the cell are very important that maintain threshold levels of CAMP. One is synthesis, one is phosphodiesterase. So if you want CAMP mediated signaling to be on, synthesis will be more and degradation activity will be lesser. Phosphodiesterase activity will be lesser. In that way, CAMP levels will be made possible in the cell to maintain at a threshold level. Understood? So that is what in the first point here, CAMPs are the second messengers, 3 prime, 5 prime cyclic AMP, and in the cell it is produced from the ATP by reaction of two enzymes as we discussed, adenylyl cyclase and phosphodiesterase. Understood? So the CAMP can influence many cellular functions. The CAMP mediated signaling in the cell is responsible for controlling the process like gluconeogenesis. Neo means new. Gluconeogenesis, new glucose formation. That is not glycolysis or this gluconeogenesis is new glucose from the non-carbohydrate sources. Okay. Glycogenesis is reversal of glycolysis. Gluconeogenesis is a process where you will have glucose from non-carbohydrate moieties. So that is during the starvation, this gluconeogenesis is responsible for formation of glucose from non-carbohydrate sources like amino acids. So that gluconeogenesis is under the control of in the cell by CAMP. Then glycolysis, lipogenesis, muscle contraction, membrane secretion, then learning process itself, ion transport, differentiation, growth control, and apoptosis. All the important physiological events of the cell are under the control of CAMP mediated cell signaling. So both enzymatic activities of adenylyl cyclase and phosphodiesterase in the cell cooperate so that CAMP gradient will be maintained, which is under the regulation of spatio-temporal effects, so that for that particular cell at that particular condition, CAMP mediated signaling will be either on if it is required and off if it is not required. Understood? So two enzymes are very important. So important feature of CAMP signaling is the co-localization of the enzymes of CAMP metabolism and the targets of CAMP. So in the CAMP signaling, if you see, because CAMP is basically a diffusible molecule, polar molecule, hydrophilic second messenger type. So it is present in the cytosol. When it has to continue its effects passing to the downstream effector, when that will happen? If CAMP can translocate to the site where you have CAMP target protein and other metabolizing agents. Okay, and that is happening in case of hydrophilic second messengers through the process of diffusion. So, for example, adenylyl cyclase enzyme which is responsible for the formation of CAMP from ATP. Then phosphodiesterase, which is responsible for degrading the CAMP, and protein kinase A, that is the effector of CAMP. So all these, the regulating CAMP enzymes and target of the CAMP, all these proteins have been found to co-localize. Co-localize means together they will be at the one place at the same subcellular site, allowing for a precise control. You will see all these three at one particular site in the cell. Why? Because that is the way CMP signaling will pass on. So precise control of CMP formation, degradation, and target selectivity is happening at a particular place in the cell. That's why co-localization is a very important feature of CAMP signal transduction pathways. So I'm stopping here and we are going to discuss further CAMP mediated signaling pathway in the next class. So any questions, clarifications if you require, you please ask me now. Anybody, if you have not followed. It should come from your side. Padmashri, Soumya, please unmute and tell me if you have followed or if you need further explanation.
Yes, sir, I follow.
False.
Okay, then I'll stop here. So I'm going to share all these slides today, and you please finish preparation of notes for your exam for the topics that we have discussed. So I'll stop here and we'll continue in the next class.